Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Tania Chakrabarty from Thermo Fisher Scientific.
Thank you for the introduction. Today, I will talk about the development and validation of a highly robust multiplex qPCR assay for the quantitation of residual plasmid DNA carrying Annamycin resistance genes that are used in bioproduction and vaccine manufacturing workflows. The assay that I'm about to describe is important in ensuring patient safety and assurance of product quality in cGMP manufacturing workflows during lot release testing, or may be used during process development and optimization. To begin, regulatory agencies require a variety of testing to address the safety, efficacy, and purity of drugs and drug products. Shown here are a few of the solutions we offer at Thermo Fisher Scientific specifically for bioproduction. On the left panel are bacterial and fungal ID products that are based on DNA sequencing methods and are used for environmental monitoring. On the right-hand side are products that utilize real-time PCR for rapid product safety and impurity testing. These include adventitious agent detection for mycoplasma and viruses, as well as residual DNA quantitation. To address the growing needs of such testing, we have developed a residual plasmid DNA quantitation assay, and that will be the focus of my talk today. So why create a simple, highly sensitive quantitation assay for residual plasmid DNA? More and more viral vector vaccines are being developed today, representing a shift in vaccine manufacturing technology. These vaccines use plasmids as starting material. mRNA vaccine technology is increasing in popularity, and DNA plasmids are the templates upon which the mRNA is built for use in such vaccines. COVID-19 vaccine is one such example. For lot release, the final vaccine product must be tested for residual DNA clearance. Another field that is showing amazing promise and therefore is growing at a tremendous rate is the field of gene therapy. Every year, there are more and more products moving forward in the development pipeline, with over 3,000 candidates currently in clinical trial. Many of these are viral vector-based therapies and must also meet regulatory guidelines for residual DNA. This slide shows the variety of therapeutic viral vector types, many of which utilize plasmids in the production process. Therefore, customers need tools and assays to measure the amount of residual DNA in their process and final drug product. One of the most common types of viral vector therapies is based on adeno-associated viral vectors, or AAV. Here, we show an example of a typical AAV production workflow. These vectors are often produced in HEC293 cells with triple transfection method using plasmids containing the rep, cap, and genes of interest. After bulk harvest, it is important to optimize the downstream purification process to reduce the amount of not only residual host cell DNA, but also residual plasmid DNA. Such process-related impurities arising from upstream processes need to be cleared for final QC and lot release of the drug product and must be within acceptable safety levels. Shown here is a specific example of a viral vector production workflow where residual plasmid DNA analysis is carried out to address process impurities such as post-harvest and viral purification, and also for QC and lot release. Our quantitative Annamycin assay kit can be directly used for such applications. Let us take a more detailed look at the regulatory guidance around residual DNA. Per FDA guidance for CMC for gene therapy IND applications published in January 2020, It says that you limit the amount of residual DNA for continuous non-tumorigenic cells to less than 10 nanogram per dose, and the DNA size must be limited to below approximately 200 base pairs. Similar recommendation can be found in the US FDA 2012 guidance. The most recent WHO recommendation also sets the upper limit of residual DNA at 10 nanogram per parenteral dose. Residual plasmid DNA is a part of the 10 nanogram per dose limit along with host cell residual DNA. The need to test residual plasmid DNA presents some challenges. Because there are not very many commercially available solutions, companies often have to develop their own assays. This is both time-consuming, technically challenging, and also labor-intensive. For example, establishing a reliable standard for a qPCR assay to be robust and meet the LOD and LOQ requirements involves time, cost, and skilled resources. In addition, there is a need to establish QC methods to ensure the assay kit is working reliably and to be able to maintain an inventory of such kits. In-house residual plasmid quantitation assays may be specific to one drug development process, and often additional development is needed when other plasmids may be used in the future. That is why we have added the ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit to our ResDNA portfolio, and this is the first one that can be used for residual plasmid DNA testing. We added this kit to the variety of already validated ResDNA-seq kits that have found extensive use in residual host cell DNA testing. Specifically, the ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit is a two-color multiplex quantitative qPCR assay. This kit can be used by gene therapy or vaccine manufacturing customers who are currently using plasmids in their bioproduction workflow. This assay is robust, easy to use, highly sensitive, and can quantitate residual plasmid DNA by targeting all commonly used kanamycin resistance genes. Just like all of our ResDNA-seq kits, our new ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit is not only an assay, but also part of a flexible and integrated workflow solution that is suitable for cGMP. The workflow begins with a sample prep, followed by qPCR using kit reagents, and finally data analysis using AccuSeq software. Once the nucleic acid is extracted and purified, it is loaded onto an applied biosystem qPCR instrument that runs the Tachman-based assay on either a 7500 fast or a QuantStudio 5 real-time PCR instrument. By leveraging Tachman technology, very high sensitivity and specificity could be achieved, and that is essential in quantitative residual DNA testing. You will see from the data presented today that the assay validation results support high sensitivity, specificity, wide dynamic range, and high reproducibility. Finally, the data is analyzed using a 21 CFR Part 11 compliant AccuSeq software for the quantitation of the residual plasmid DNA that has the ability to maintain an audit trace. So let's start with the introduction to the product concept before diving into the validation study design and sharing the results from those validation studies. The ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit offers a two-color multiplex assay that targets all common alleles of the kanamycin resistance gene found in plasmids used in bioproduction today. The highly sensitive quantitation via real-time PCR is achieved by using proven applied biosystems Tachman real-time PCR technology. The assay uses the FAM dye to detect three conserved regions among the different kanamycin resistance gene families. The NED IPC acts as the internal process control to verify the absence of inhibition during PCR amplification. To achieve required sensitivity, a DNA-free master mix was utilized. The kit includes innovative DNA-free master mix, triplex Tachman primer probe mix, and plasmid DNA standards. The assay is easy to use and offers results under 5 hours. The high sensitivity and specificity of the assay has been achieved by using an innovative DNA-free master mix. In order to validate this assay, for residual plasmid DNA testing, we focused on the following performance parameters, such as PCR efficiency, linearity, dynamic range, LOD/LOQ, accuracy, precision, specificity, and PrEP-seq spike recovery. We validated the performance of this assay using multiple users, specifically 10 operators, spanning multiple days, and using multiple qPCR instruments such as the QuantiStudio 5 and ABI 7500 Fast. These experiments were run using multiple modes of extraction, namely manual and automated, and were carried out across multiple sites in the US and abroad. In the next few slides, we will cover results from the validation study that establishes the robust performance of the ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit. Shown here is the standard curve performance data collected across 25 qPCR runs using 2 lots of reagents and run on 5 different QuantStudio instruments. The calculated PCR efficiency met the requirement of 100 plus minus 10% and an R-square greater than 0.99, thereby demonstrating high linearity and efficiency to enable quantitative results across a broad range of DNA concentration. The results shown here are from a large number of reactions that demonstrate high overall precision down to an LOQ of 30 copies per reaction. As you can see, the IPC-CT remains steady across a wide range of kanamycin plasmid concentrations that were tested down to 15 copies. The assay is highly precise, and the performance is robust as measured by multiple operators, spanning multiple days and across multiple instruments. Another product requirement we met is the spike in recovery rate of the plasmids from DNA extraction using both manual and automated workflows using PrEP-seq kit. The results shown here demonstrate that the specs of 70 to 130% recovery rate were successfully met and were consistent across both the extraction workflows. The results shown here demonstrate a clean separation in CT values between the LOD/LOQ of 30 and 15 copies and the NTC. This will enable customers to meet regulatory requirements for measuring very low levels of residual plasmid DNA containing kanamycin resistance genes in order to meet regulatory requirements. We monitored the CT of the IPC and the kanamycin target genes in a variety of different matrices that included benzonates, detergents, and cell culture media post-sample prep. These matrices are representative of highly inhibitory substances commonly found in bioproduction workflows. The robustness of the IPC-CT values is indicative that our kanamycin assay performs well even in the presence of common inhibitors. Last but not the least, we tested the kanamycin resistance gene residual plasmid DNA assay performance against a variety of exclusion panel substances to ensure there is no cross-reactivity. The IPC amplification remained the same in the presence and absence of the variety of cross-reactants, demonstrating that our assay is highly specific. In conclusion, our results demonstrate that the ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit could detect its targets quantitatively in a variety of matrices corresponding to workflows that are used in gene therapy and vaccine manufacturing. This residual plasmid DNA assay is compatible with both manual and automated extraction workflows using PrEP-seq kit and gave robust results on both QuantStudio 5 and ABI 7500 real-time PCR platforms. The assay is highly sensitive, LOQ of 30 copies, LOD of 15 copies, and also specific, showing no cross-reactivity as tested using exclusion panels. providing validation data like these demonstrate that the kanamycin resistance gene assay is highly sensitive, specific, and robust. The all-inclusive reagent kit containing plasmid DNA standards will give our customers a valuable solution for lot release testing that can be carried out in less than 5 hours. Assays such as this will help ensure patient safety by quantitatively measuring residual plasmid DNA in the final drug product used in gene therapy, vaccine manufacturing, and other biotherapeutics workflow. And last but not the least, thank you very much for your attention.
Okay, thank you. So, our first question for our speaker is, you mentioned this assay can quantify all plasmids containing a kanamycin resistance gene. With various kanamycin resistance genes that exist, how is this achieved?
That's a great question. So we analyzed a huge number of kanamycin resistance alleles that are commonly used in bioproduction and looked at various kanamycin resistant gene families and finally designed a multiplex assay that targeted the conserved regions for the multiple gene families. With this approach, we are quite confident that this is one assay that can cover the vast majority of the kanamycin resistance gene alleles that are out there.
And do you have any other related residual impurity testing kits for gene therapy workflows?
Yes, there are other ResDNA-seq flavors that might be relevant for gene therapy workflows, some of which are the ResDNA-seq HEC293 kit, kits for E. coli, as well as the simultaneous quantitation of SF9 host cell and baculovirus DNA. We are considering developing additional qPCR assays to quantitate residual plasmid DNA with other antibiotic resistance genes such as ampicillin, but chose to start with kanamycin resistance gene because of its prevalent usage in the field. We also offer custom solutions should you have special needs. Please contact a Thermo Fisher representative to learn more. So please let us know what your needs are and we can address them.
And is the DNA standard included in the linearized or supercoiled plasmid DNA?
The DNA standard included in this kit is well characterized, QC'd, and it's a linearized plasmid DNA.
I see the results are given in copy number, but the regulatory guidance is given in mass. How do you convert copy number to mass? Is this done in the AccuSeq software?
AccuSeq also reports results in copy numbers, and we need to be converted to mass. The mass will be dependent on the size of the plasmids used in your process, but can be measured, and this conversion has been validated. When using multiple plasmids, such as the triple transfection production of AAV, we recommend to use the average mass of the 3 plasmids as a conversion factor. Our field application specialists can assist you if necessary.
How does the assay deal with matrix inhibition, and will you be able to tell if there is a PCR inhibition due to matrix effects?
Great question. Yes, the assay provides 2 methods to assess the presence and extent of the matrix interference. The first one is an internal process control, or the IPC, that is running in the background and will provide a first assessment if there is PCR inhibition. The second measure is the utilization of spike recovery experiments. We also have an experienced team of field applications specialists to assist with the implementation, qualification, and validation of this assay and any challenges that might occur along the way. So please reach out to them.
Okay, and what types of sample matrices were used in the validation study?
We have used pseudo matrices from the harvest, purification, and final bulk stage of a typical gene therapy process, and have included additives like benzonase, excess of unrelated DNA, detergents, and typical cell culture media.
Okay. Is it possible to use the ResDNA-seq quantitative plasmid DNA kanamycin resistance gene kit successfully without DNA extraction?
So yes, we have validated this kit using samples post-extraction, but some of our customers have had success using this kit even without extraction at the QC and lot release stage. However, we recommend if you're going to use this kit upstream for assessing process impurities, then you must do a DNA extraction prior to running the qPCR test.
Okay, thank you so much, Tania.
Thank you very much.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.